Dear students, in this video we are going to discuss about renal concentration test. So renal concentration test is used to measure the tubular functions of the kidney. So it is based on the ability of kidney to concentrate the urine. These are that is proximal convoluted tubule, loop of Henle, and then distal convoluted tubule. So these are all the various parts of nephrons and their uh, function, that is reabsorption of concerned metabolite. So the first one is proximal convoluted tubule. So in this part of nephron, it can reabsorb the glucose, amino acids, uric acid, sodium, chloride, water, and then and also secretes hydrogen, and also it maintains the acid, base, and then ammonia. And then next one is loop of Henle, that is it can reabsorb the sodium, chloride, magnesium, and then calcium. And then next one is distal convoluted tubule. So in this region of nephron, reabsorbs water, sodium, chloride, that is electrolytes. It can reabsorb water, electrolyte, uric acid, ammonia, and also secretes H+, that is hydrogen. And then the important topic that is used in this frequently used in this topic is, that is renal threshold. So renal threshold is defined as the plasma level of the glucose is excreted in the urine. That is the normal renal threshold of glucose is 160 to 180 milligram dl. If exceeds the 180 milligram per dl of glucose concentration, obviously the glucose will be excreted in the urine. So that condition is called as glycosuria. And also to assess the renal tubular function of kidneys, to determine the specific gravity of the urine, and then concentration test. The concentration test of renal tubular function is to determine the specific gravity of the urine. And then last one is PSP dye test. So these are all the various tests of renal concentration test. And then specific gravity. So specific gravity is the index of concentrating ability of the renal tubules. So the normal specific gravity of urine is 1.02. If this specific gravity is the urine specific gravity is increases during the condition of dehydration and apnea mellitus. And then the specific gravity level is decreased in the renal failure and also diabetes insipidus. And then next one is, that is urine concentration test. So there are three types of tests to determine the ability of kidneys to concentrate the urine. Three types of tests to determine whether the kidneys are properly concentrate the urine or not. So that is, there are three renal concentration tests or urine concentration tests. So the first is Fishberg concentration test. So this test is frequently used. Next one is Lashmet and Newberg concentration test, and then third one is posterior pituitary test. So Fishberg concentration test. So this test imposes less strenuous condition, curtailment of fluid intake. So this Fishberg concentration test is a highly fluid restriction for 24 hours. So the procedure for this Fishberg concentration test is, the patient is allowed no fluids from night to next morning. The meal is given at 7 p.m. That is, the evening meal is given at 7 p.m. So it should be a protein, that is highly protein, and must have a fluid content of less than 200 ml provided for the patient. And then urine passed in the night is discarded. Nothing by mouth taken by next morning. Collect the urine specimen for 1 hour interval 3 times, for 1 hour interval for 3 times, and determine the specific gravity of the urine. So Fishberg concentration test is frequently used, and also 24 hours fluid restriction for this test. And then interpretation of this Fishberg concentration test is, if the tubular function is properly functioning, if the tubular function is normal, then the specific gravity is 1.02. if the tubular function is impaired with some other diseases, because some other diseases means it can't uh, work properly, it can't function properly. So the specific gravity of urine is 1.02 or less. In severe renal damage condition, the specific gravity is less than 1.02. And then next type of renal concentration test is, that is Lashman and Newberg concentration test. So this test imposes severe fluid intake restriction over a 38 hours and involved in the use of dry diet for 1 day. This test is not frequently used because um, more than 3 days for fluid restriction is needed for this Lashman and the Newberg concentration test. That's why only this test is not done by the patient. So and then third test is, that is posterior pituitary extract. So posterior pituitary secretes a specific enzyme called posterior pituitary um, posterior pituitary secrete a specific hormones is, that is vasopressin. So the subcutaneous injection of 10 pressor units of posterior pituitary extract, that is vasopressin, in a normal person will inhibit the diuresis produced by the ingestion of 1600 ml of water in 15 minutes. And the interpretation of this posterior pituitary is, under the condition of this test, individual with a normal kidney function excrete urine with a specific gravity or higher. Failure to concentrate the urine indicates the renal damage. And then last test of this renal concentration test is PSP diet test. So this PSP diet test is also known as 15 minutes PSP diet test. Why they named as 15 minutes PSP diet test means, because this PSP is, that is phenol sulfathalin diet test. So this dye is injected in the patient through IV. Then this PSP will be excreted, that is half of the nearly 30 to 50 percentage of dye should be excreted in the urine within 15 minutes. So that indicates the proper functioning of kidney, that indicates the normal functioning of kidney. That's why only this PSP dye test is also known as 15 minutes PSP dye test. So the use of PSP, that is phenol sulfathalin, is to measure the renal function. It was first introduced by Rountree and Granacki in 1912. Later on, Smith has shown that the amount of dye employed, 94% is excreted by tubular function and only 6% is excreted by glomerular filtration. That is, the 100% of dye, the nearly 94% of dye should be excreted only by the tubular function of the kidney, and remaining 6% of the dye should be excreted in the glomerular filtration. And then it is the dye of choice of excretory function of kidney, that is a specific dye it to choose only for to determine the excretory function of kidney. And then it is non-toxic, and 94 percentage of um, dye, that is PSP dye, is excreted by tubular secretion. And then procedure and interpretation for this PSP dye test. So it has been shown that this test is reliable and sensitive if the amount of dye excreted in the first 15 minutes. So already I told in the first previous slide, that is PSP dye test is also 15 minutes PSP dye test. The PSP dye excreted within 15 minutes indicates the proper normal functioning of kidney. When 1 ml of PSP phenol sulfathalin dye is injected by IV, the normal kidneys will excrete 30 to 50% of dye during 15 minutes. So nearly 50% of dye PSP is excreted within 15 minutes. That represent the normal functioning of, that is normal tubular function of kidneys. The amount of dye is excreted in 2 hours indicates impaired renal function. That is, the time of dye taken for 2 hours to excrete the whole amount of dye, that indicates the renal problem. That is renal tubular problem. Uh, so the amount of dye is excreted in 2 hours indicates the impaired renal function. Thank you.